Krzewinski, and I'm the stranding technician for the Lowcountry Marine Mammal Network. To begin my journey to become a stranding technician, I first started by getting my bachelor's degree in marine biology from Hawaii Pacific University. This was a long way from where I grew up in Green Bay, Wisconsin, but I wanted to pursue my passion for marine biology. While in Hawaii, I always had a passion for marine mammals, so I decided to pursue this by becoming a volunteer with the University of Hawaii's Marine Mammal Stranding Program. As a volunteer, I got to help with lots of different necropsies, seeing a bunch of unique marine mammal species that I didn't even know exist up until then, and do other lab tasks like helping sort microplastics and doing diet work. After this, I then knew I wanted to pursue a higher degree, so I then went to the University of Miami's Rosenstiel School of Marine and Atmospheric Science to obtain my Master's of Professional Science in Marine Mammal Science. Part of this program, I had to complete a internship, which I did with Florida Fish and Wildlife Conservation Commission Southwest Field Laboratory in Port Charlotte, Florida. As an intern, I got to help with manatee and cetacean rescue, release, carcass salvage, and necropsies. As an intern, I also had to complete a research project for my master's, and I did this by building an otolith reference collection to be used eventually for cetacean diet work there. One of my primary interests in research has been diet work. Diet work is very fascinating to me because we can learn from dead, stranded animals what they're doing then in the wild, and it can all help for conservation and management. If we know what they're eating, then we know where they are living a little bit. By doing this, we look at the otoliths, which are the fish ear bones. These are actually unique to each family and then further to each species. So you can look at them, identify them, and then retrospectively figure out what that animal was eating. It's a very tedious process. First starts by sorting the stomach contents that we collect the stomach hole from during necropsy. Once you then have the otoliths, you have to hand sort them. I typically sort them by ones that are similar in shape and size, and then I have to look under the microscope, identify them, count the number, to then get a minimum estimate of the number of fish eaten. For example, I can say that one dolphin consumed 5 Atlantic croaker, 2 star drum, and 1 striped mullet. All of this again just by looking at those otoliths, identifying them, and counting them. One of my research interests is dolphin diet. To do this, we first collect stomachs whole during necropsy, and then we will sort the contents over a sieve, have to pick through it by hand to separate out otoliths, which are fish ear bones, and squid beaks. Here I have some squid beaks that have been sorted from stomach contents, and here are some otoliths. Once the squid beaks and otoliths have been separated out, I let them air dry for a little bit, and then I go through and count and identify them. In order to do this, I have a microscope like pictured here, my forceps, and usually a petri dish to help sort them out. In order to identify fish otoliths, you have to look at a surface on their structure called the sulcus. And it's actually the nerve groove, as it's their fish ear bones, and each species has a unique shape and structure to the sulcus. In addition to that, each otolith actually has a unique shape. So there's many different identifiers that you can use in order to identify the otoliths. It's really nuanced, really detailed, and to do this, you need to look at reference otoliths, or some that have been previously identified like we have here at the lab. Compare them in order to make your best guess as to what that otolith is. So first, I'm going to start sorting my otoliths, looking at ones that are similar in shape and size. So for example, you can see that these here all have similar shapes and sizes. So I'm going to put them to the side because I think that they are all the same species until I look under the microscope. Next, I'm going to turn my microscope on, and I'm going to lay all the ones that I think are the same ones under the microscope. And I'll do this by laying them left versus right otolith, as they actually all have pairs. So one of the ways that we do this is we look at the orientation of the sulcus. If it faces one way, it's usually the left otolith, and if it faces the other way, it's the right otolith. So now I have all my otoliths sorted under here, the microscope. I have my left ones laying out on the left and my right ones laying out on the right. I looked at all of these under the microscope and was able to confirm that these are from Atlantic croaker. So my next step now is going to be counting them. So I see we have 1, 2, 3, 4, 5, 6. 6 left otoliths, 1, 2, 3, 4, and 4 right otoliths. I'll make a note of that on our data sheet, and then finally I'll say how many Atlantic croaker that this one specific dolphin did. Now I do this by looking at the greater number of left or right. In this case, we have 6 left otoliths, so we can say at minimum that this dolphin consumed 6 Atlantic croaker. I still have some otoliths left here, so I'll keep looking at them under the microscope, counting them, looking at them from left and right, and seeing how many fish overall that this dolphin ate. Currently, I am working on a paper looking at diet of bottlenose dolphins in South Carolina waters from 2008 to 2015. One reason we did this time frame is because we wanted to look specifically at 2011. This was a very cold winter, we had really cold water temperatures, and as a result there was a bottlenose dolphin unusual mortality event. We thought that preliminary prey items of dolphins, common ones that they were consuming like stardrum, Atlantic croaker, and silver perch were dying, and that way they were having to shift their prey to other non-preferred items, specifically hake. We were able to confirm this by looking at stomach contents. And this paper also will have other cool things looking at diet comparisons between seasons, historically and currently, and other things like gender and human interaction classifications. As a stranding technician for the Lowcountry Marine Mammal Network, my job entails coordinating stranding response across the entire state of South Carolina. We respond to 50 to 70 strandings each year, most of which are Atlantic bottlenose dolphins. Typically, I report to the lab every day, and then if I get a call, I'm responsible for calling that caller and formulating a plan. This could be as simple as going to pick the dolphin up off the beach, or if it's in the marsh or somewhere inaccessible by land, I have to figure out a way to get out there by boat. Once that happens, then we either get to the carcass or collect the carcass and bring it back to our lab, and then conduct a necropsy. A necropsy entails collecting a lot of samples, usually looking at every organ and describing it, looking for any abnormalities and significant findings to hopefully try to determine why that animal died. After the necropsy, we do different things then, like storing our samples permanently in our freezers, or sometimes I'll be sending off samples right away depending on the sample type to get analyzed and hopefully figure out that cause of death. Also, each necropsy entails lots of cleanup. So this would be putting samples away, cleaning our necropsy table, cleaning all of our supplies, and then restocking that bag so we're good to go for the next stranding response. During a necropsy, we collect many different samples. However, what samples we are able to collect depends on the decomposition state of the animal. Typically though, we always aim to collect standard samples, which include skin. This is used for genetic testing. Teeth, which are used to age the animal, because on cross-section they have rings like a tree and you can count the rings to age the animal. Lung and brain to do both viral and bacterial testing. The stomach and intestines for diet and microplastic analysis. And the reproductive organs, because then we can figure out if that animal was sexually mature. And then using all the other information from that animal, we can look at the size that animal is mature and how old they are when they are mature. If the animal is really fresh, we are able to collect histocassettes of every single organ, which are really small ones that get fixed in formalin, and are then sent off to a pathologist. Typically, we do this for every single organ, ranging from every single lymph node, the lungs, the brain, the kidneys, and the liver. Other samples that we can collect if the animal is fresh is blubber for contaminant testing, and liver and kidney, which is also used for contaminant testing, among other things. Sometimes for strandings, we will also collect skeletal parts, specifically the skull and sometimes vertebrae. This is used then for life history. We will clean up that skull, do a bunch of measurements afterwards, and then can look at different things like if vertebrae were fused or not, and how old that animal was, and then if it's a certain species or not. Once each necropsy is completed, we've cleaned up, we've put those samples away, we've sent off the relevant ones, then we have to write a report for each animal and upload photos, create a database entry, and do lots of paperwork just for one event. My other lab duties entail doing skeletal cleaning. There will be another video detailing this, but first you have to flense the part, then you put it into our beetle colony and we bleach and degrease it, and then it gets measured and ultimately sent to the museum in Columbia.
How does responding to a dead animal help us protect the ones that are still living?
Responding to dead stranded animals is really important because we can identify different threats that are impacting these animals. These can be things like human-related activities. Say if an animal is getting entangled in a crab pot, then perhaps we could lead to different gear suggestions to prevent that animal from becoming entangled. And also then we can identify different threats like diseases. We need to understand what's killing these animals and harming them in order to protect their wild counterparts better. Here at the lab, we store our samples a few different ways, one of which is frozen in our negative 80 degrees Celsius upright freezers. We typically do this for biological samples like the lung, brain, kidney, liver, and blubber. When they're kept this cold, they are still used for many different things, and then if somebody along the way comes back and they want a sample, we're able to pull it from our freezer and send it to them. We have samples in our freezers dating back to 1987, and many different species like bottlenose dolphins, pygmy sperm whales, dwarf sperm whales, stinellas, among other species. Other samples that we keep for a long time are kept in ethanol. Typically, these are our reproductive samples, so the testes or the ovaries, which we can eventually do reproductive analysis, determining if that animal was sexually mature or not. So we have really big cabinets that are flammable and meant to keep that ethanol and keep those samples safe since they are in chemicals. Other samples are kept in our really big negative 20 Celsius degree walk-in freezer. This is typically our skeletal parts that we will collect during necropsies like skulls, ribs, vertebrae, and then also our stomach and intestines. These are our flammable cabinets to keep all of our samples that have ethanol. These can be some fully preserved organs like hearts, believe it or not, all the way down to teeny tiny testes or ovaries. Let's take a look at some. For example, we have this testis here from an animal that we will eventually examine to figure out if this animal was sexually mature or not. Alright, this is our freezer room. As you can see, we have 3 upright freezers and then 1 chest freezer. These are all at negative 80 degrees Celsius to keep the samples super cold and preserve them. Let's take a look at what we have inside some of them. Making sure we have our thick gloves on so our hands don't get burned from the cold. We keep all of our samples organized here in racks, which are numbered and organized. And then for each rack, we can have different samples. Like this one is full of blubber samples, for example. Here is one of our largest freezers that we have here at the lab. It currently has over 200 samples in it, most of which are large skeletal parts and stomach and intestines. Dolphins typically face 2 different kinds of threats, category-wise. The first is natural causes, and the second is anthropogenic or human-related causes. Some of the common natural causes that we see here in South Carolina as to why dolphins strand are disease. Commonly here we see brucella, which is a bacterial infection, generally passed down from mother to calf, and it can cause abortions. But this is typically a bacterial infection that can impact the lungs and the brain. Other natural causes are things like old age, predation, or sometimes, specifically in the spring, we have calving season, but unfortunately many of these calves don't survive. They can be stillborn, they can be really heavily contaminated as moms pass down their contaminants to their offspring through their fat reserves. Or sometimes it can just be a stillborn that maybe had genetic defects. A stillborn that had genetic defects and ultimately was born as a stillborn. We also see anthropogenic or human-related causes. These really fluctuate annually how many we see, but typically, historically, our averages has shown about 25% of our strandings are due to anthropogenic causes. The most common of which are entanglements and crab traps. The most common entanglement that we see here is in crab trap buoy lines, typically from the Atlantic blue crab fishery. Sometimes this will be the animal's curious getting too close, other times it's derelict gear that the animal happens to get stuck in, and that buoy line will get wrapped around it and keep constricting as the animal swims with it. You can help protect dolphins too by reporting any strandings that you see. We rely on reports from the public in order to respond to dead or alive dolphins. For live ones, we ask that you do not touch the animal. Do not push it back, do not intervene or try to disentangle it. Instead, call us immediately and we will get our ball rolling and figure out what we need to do to respond to that animal and get the proper permission from authorities. If you come across a dead stranded cetacean, again, please call that SCDNR Wildlife Alert Hotline. It'll get connected with us and we will talk to you and we'll formulate a plan for how to get there. We always ask, depending on the situation, live or dead, for pictures and especially for those live animals videos. So you can do your part by keeping your distance, reporting it, getting that information, and waiting until we get in contact with you, can ask you the questions that we need, formulate a plan, as we may need your help. But only, again, help us if we ask you specifically to. But regardless, we always appreciate the reports. One of the ways that you can help dolphins is by engaging in proper boating activities. One thing that we want to do is especially obey those no wake signs and go slow in certain areas where we know dolphins are common. If you do see a dolphin on the water, please shift to neutral and keep your distance. It's recommended to stay at least 50 yards away from dolphins. This way, sometimes animals are curious, they will come up to you, but this way it can prevent them from getting injured and sometimes even dying, say if they were to get struck by that boat. Other things that you can do are remove your marine debris from the water. This includes fishing line, nets, or if you see any in the water, safely get it out of the water to the best of your ability. This can prevent things like entanglements. Sometimes it will be a discarded trap or line that the animal will then get life-threatening injuries and sometimes even ultimately dead strand. So with these ways, you can protect the dolphins in our waterways. Some of my advice to anyone who wants to become a future stranding biologist is to volunteer or intern. Through volunteering or intern, you get a sense of what it is to be a stranding biologist and the long, demanding days that it entails, but also you learn so much. You can learn the different terminology, the different roles, and just really get a sense of what a necropsy entails, as it's not for everyone what the sight and smells. Along with getting involved, don't be afraid to ask for specific roles or to ask questions. I learned so much just by asking questions during necropsies and figuring out what's normal, what's abnormal, and then also asking about different projects that I can do to become involved. This way I really developed a good research background and figured out what my specific research interests were, which were really helpful in grad school.